All right, let's get started. Ladies and gentlemen, good afternoon, good morning, and good evening to you all, and welcome to Season 2 of the Space Generation Advisory Council webinar series on health in space. We are at the start of a new episode that's all about genes in space. My name is Jules Ancet, and my guests of today are Dr. Tricia L. LaRose and Dr. Francine Garrett-Bakelman. Again, welcome back to our series. Of course, venturing out into deep space will give rise to new challenges, not only to the technologies used, but also to the human element in this type of journey. Our bodies and minds will be put to the test when we travel to Mars on a 3-year return journey, or even in the next human missions to the moon. And today we will talk more about some of the health and life science research around genetics and cell biology in space that supports these efforts, and ultimately about what experiments out there teach us about health here on Earth. It is a topic of great interest to students, young professionals in terrestrial and space fields, medical doctors, engineers, and other space health enthusiasts. We've seen many registrations, and in fact, we're fully booked today. And we invited 2 great experts for our episode today. I will introduce our 2 speakers now, and then after their introduction, I have a little housekeeping announcement. And then finally, we'll start with their presentations. We'll hear today from Dr. Tricia LaRose. She is the principal investigator for tumors in space with the University of Oslo in Norway. Her cancer research project has been selected by the United Nations Office for Outer Space Affairs and the China Manned Space Agency for a 31-day space mission on board the China space station to be launched in 2025. Beyond her academic work, Tricia works as a medical advisor at the Institute of Aviation Medicine for the Norwegian Armed Forces Medical Services and as an adjunct lecturer at the International Space University. She is also a scientist astronaut candidate with the Project Blossom, a children's book author, and a motivational speaker. And after Dr. LaRose, we will hear from Dr. Garrett-Bakelman. She is an assistant professor in biochemistry and molecular genetics at the University of Virginia in the USA. After studying biochemistry and cell biology, Francine continued on to study medicine at the Albert Einstein College of Medicine in New York, where she attained her MD and PhD. Afterwards, Francine specialized in epigenetics, hematology, and oncology. And besides growing her expertise in the medicine and life science field, Francine worked closely with NASA on the famous twin study, where one of the twin astronauts, Kelly, spent a year-long mission on the International Space Station. Therefore, Dr. Garrett-Bakelman and her colleagues were able to examine molecular and physiological traits of astronauts that may be affected by prolonged time in space. We'll get you to Dr. LaRose and Dr. Garrett-Bakelman shortly, but first, a few housekeeping items. You have been muted upon entry to avoid background noise and disruptions. This session will be recorded for later viewing. And this webinar, as it is a feat of engineering, is not comparable to anything being accomplished in space. However, this is still a global effort with your speakers and moderator being in 2 different time zones already, not counting you, the audience. Of course, we are not counting on any disruptions, but Murphy's Law does apply. And in that case, please bear with us as we try to restore the live session as quickly as possible. And finally, feel free to use the chat box during the presentation for questions and comments. And during the Q&A part at the end of this session, I will ask your questions to our speakers. Now, I've been talking a lot. Let's get to our first speaker, Dr. Tricia LaRose. Hi, Tricia, and thanks so much for being with us today. I'll be switching over to you now, if you don't mind to share your screen.
Hi, hello, good morning, good afternoon, good evening. I'm just setting my timer so I can be sure that our second speaker, Dr. Professor Francine, has enough time. I don't want to go over time, so just give me 1 second. There we go.
Excellent. I found out that we already spoke about this in October 2019. I'm happy that we made this work today. I really appreciate you doing this. The virtual floor is yours.
Wonderful. I'll share my screen. Let's see. Hi, everyone. I'm going to be completely honest with you. I'm in a strange mood. I'm feeling very philosophical and very creative. So let's see where this presentation goes. It might go off on a few tangents, but thank you, Ivy. Thank you, Jules, for inviting me. It's great to be here. Do answer some questions. So as I said, our Dr. Professor Francine with the NASA Twin Study is coming up after me, a molecular geneticist, and they have tons of beautiful data that I'm sure she's going to share. So I thought today, to start this off, because this is the SGAC, we have students, we have early career researchers. I want to give you a quick overview of tumors in space, what we intend to do. We obviously don't have results because we haven't done our space flight, but we are starting our pilot experiments here on the ground. But I thought what I would share with you is a little bit more of the pathway from idea to realization of a space experiment, specifically in biology, specifically on organoids, and specifically related to genes in space. So as Jules mentioned, tumors in space has been selected by the United Nations and the China Manned Space Agency for a 31-day space mission. So sprinkled throughout this presentation, you're going to hear a lot of talk from me about geopolitics, about funding, about challenges. I mean, what we've designed with tumors in space is a perfect experiment. When you sit down and you write methods and you write experimental workflows and you optimize every step along the way to the point where you have perfectly matched cases and controls, your timing's perfect, your batch samples are perfectly matched, you have all the funding you need, all of your partners sign on the dotted line, there's no time delays, there's no geopolitical issues, this is what we've designed. But that's not what happens. And so what I want to share with you is some of the things that come up in designing these quite sophisticated experiments in space that never make it into a peer-reviewed publication. So I'm really kind of laying myself out here. I'm going to talk about my failures and talk about my lessons learned and give you a little bit of input into behind the scenes of genes in space. So I'm showing this map here because this is a map of the participating states within the European Space Agency. So our Dr. Francine is going to talk from the NASA perspective, and the NASA perspective is very, very different than the ESA perspective. Also myself as a researcher coming from Canada, my birth country, now Norway, I mean, I have my own kind of ideas of social welfare states and constructs around financing and fair distribution of resources and how our research in space ought to be applied to the benefits of human on Earth. So keep that in mind when I'm talking through my presentation. So here is a map of the ESA participating states, and you see Norway here. It's the dark season in Norway. It's very dark here, very miserable. We have 5 hours of daylight where I am. Terrible, terrible. But anyway, Norway is super well known for satellites. We're well known for, we have some sensors on the Mars rover right now in collaboration with NASA, some of my colleagues at the University of Oslo. Really satellites in defense is our strength. And so to try and pull off a space medicine experiment in Norway, where there's 5 million people and there's no space medicine portfolio, it's really, really hard, particularly because Norway is such a small country. And that means that what we contribute to the European Space Agency in terms of dollar amounts, euro amounts, Norwegian crowns, is actually quite a bit less than some of the larger countries. And how the European Space Agency works, okay, and how the funding works for tumors in space is that here I am in Norway. I have to be grounded with my national space agency. My national space, we do have a space agency in Norway, just to let you know. Our national space agency has to get behind this project, has to champion this project, internally approve it, and then send it over to ESA. So then we go through another round of evaluation, and then ESA finally releases the money. So this is a European Space Agency funded project with the support of the Norwegian Space Agency, but our funding is limited. So it really depends what country you come from and how much money is available within the specific portfolio, the PRODEX program that ESA uses to fund this type of research, whether it be biological research or any research on platforms such as the ground platforms, the parabolic flight platforms, and the ISS. So our funding here in Europe is slightly more complex than the funding that we have in the States, for example. So, So here in the center, you see an artist depiction of organoids. So one of the things that's unique about Tumors in Space is that we're using patient-derived organoids. We're not using astronauts. We're not using cell lines that are two-dimensional in nature. We're using organoids that are three-dimensional in nature that mimic the structure and function of the host tissue. And these organoids are actually derived from stem cells that are taken out of biopsies from real cancer patients. So we have a collaborator in the Netherlands who has a biobank of tissue biopsies of tumors from real patients who have given their consent. So we have real tissue that's three-dimensional in nature, that's stem cell based, that we're sending up into space. And we have cancer tissue and we have healthy tissue. So this is really interesting and really unique because any results that we get are as close as to the human body as we can possibly get without it actually being an astronaut. And as Dr. Francine will tell you, using astronauts in space is a whole other level of complexity, and the sample size is really small. But with organoids, we can increase the sample size whilst simultaneously increasing the physiological relevance of our results in comparison to animal models or cell lines, for example. So when we go into space, of course, we have the cosmic radiation, we have microgravity that we're interested in terms of the environmental factors, and we have to be able to capture both of those exposures in a meaningful way. And part of this kind of process going through the United Nations is that the research that was proposed had to never been done before, had to be completely unique. And it had to be relevant both to space and to the ground, which most space projects are nowadays. But we really had to be competitive. Think of the most, shall I say, extreme experiment that we could do whilst maintaining the scientific and logistical feasibility of our project. And so what we're doing is we're sending these patient-derived organoids into space. We have a 31-day space mission. We're looking for the unique mutational signature of cosmic radiation. We're looking to see if microgravity can slow or stop the growth of these organoids due to gene expression changes. And we see this in cell lines, but we haven't yet done this in organoids. So this will be one of the first studies. So it's very exciting. And whatever we find, we know is incredibly relevant to the human body because this is actual biopsies taken from the human body. But that's not enough. We also have to do preparatory work on the ground to make sure, I mean, as you can imagine, space experiments are expensive and they're rare. And we need to make sure that everything is working as it should be in our logistics, our shipping of the organoids, our cultivation, our pipelines for bioinformatics, statistical analysis, the genomics facilities, anything you could possibly think of, the export control measures, collaborations signed, bilateral agreements signed, everything has to be done before we can, in fact, the hardware has to be designed. This is a new experiment. The hardware, the spaceflight hardware doesn't exist on the China space station. It doesn't exist on the ISS either. So I'll come back to that. So we have to prepare our experiment for space by doing our ground-based experiments in parabolic flight. But we also have to ensure that our results, that our effort, that our research portfolio is in fact somehow designed and will yield results that can benefit people on Earth. So obviously this is a cancer experiment and there's no cure for cancer. I'm not saying we're going to find a cure for cancer, but whatever we do find will help solve some of the questions. So there is this benefit to Earth that is really important. So this is what you see on the slide. You see cosmic radiation in space, microgravity in space, and you see ionizing radiation on Earth and heavy ion radiation. I didn't put that. There's also heavy ion radiation where you can look at different types of radiation sources that are more closely related to cosmic radiation, and also the gravitational continuum on Earth. We have excess G forces during launch and descent, re-entry. We have, transportation issues with vibration and mechanobiology we have to look at. But we're also really interested in looking at to find the mutational signature of the gravitational force by increasing our G force using different centrifuges. So this is kind of the package of tumors in space. So what is a mutational signature? Here I'm showing you something that comes from Cosmic, which is the Catalog of Somatic Mutations in Cancer by Mike Stratton in the UK. They have a large repositorium of these somatic mutations in human samples. And so here I'm showing you a unique fingerprint in the actual rDNA. So mutational signature is a unique fingerprint found in our DNA. And we can find these unique fingerprints when we look at different types of exposures. So tobacco has a unique mutational signature. UV light has a unique mutational signature. All of these different carcinogenic or cancer-causing exposures have unique mutational signatures. Not all mutations are bad. Some mutations are neutral. Some mutations might have a downstream effect that are actually positive, but I think our next speaker is much more qualified to talk about that. But what I'm showing you here is that when we're looking in the DNA of our organoids after we expose them to cosmic radiation, for example, heavy ion radiation, ionizing radiation, we're able to find a unique mutational signature, a unique fingerprint of that exposure. And what's really interesting is we can start lining up that same fingerprint, that same mutational signature with different types of cancers. So here we're seeing this mutational signature of tobacco in lung cancer. And you can see here on the bottom, okay, you have UV cases up top and you have tobacco cases on the bottom. You can see here these same mutations, the C to A and C to T mutations are occurring in lung cancers. And we can see that the C to A mutation is actually found as a mutational signature in tobacco. So we can obviously see by this that we have this overlap of mutational signatures in tobacco and in lung cancer, and we see a causal effect. So we want to do something similar to in our organoids, looking at cosmic radiation as an exposure. So we're looking to find the unique mutational signature of cosmic radiation in our organoids after a 31-day mission to space. And we're looking on the ground at heavy ion and ionizing radiation in the same samples of organoids that are going to be cloned, okay? So we're controlling them for this sort of heterogeneity, this sort of issue. We're making our samples as homogeneous as possible so that when we compare these different types of radiation, we can compare the different types of mutational signatures to see if they overlap or if they're similar in some way. And when we find these mutational signatures, we can then start taking samples of blood, for example, in astronauts or commercial astronauts, for example, and look for those mutations as early detection of disease, whether that be cancer or cardiovascular disease, etc. So this is one of the things that we are doing that's kind of interesting and perhaps a little bit easier to understand for our group here. And then, of course, we're also looking at gene expression changes due to microgravity in our organoids, but unfortunately don't have time to go much into that detail. So here's I'm just checking the clock. Here's a slide showing you just that we're using organoids. There's many, many different types of organoids. We're using colon organoids, intestinal organoids, and that's because they're quite simple to use, and these are pilot experiments. But we also have access to retinal organoids, which would be super interesting because we know astronauts have incredibly an incredible risk for different types of eye diseases, because not just of the microgravity, but of the radiation. So we can start looking at different types of cancers and different types of organoids over time using our organoids. So this is to show you, I talked a little bit about the ground-based platforms that are absolutely essential to prepare for our space flight. So here on the top left-hand corner, you have a parabolic flight, which gives you, 22 seconds of microgravity in a series of parabolas, and then you have 3 flights in one campaign. So we have our upcoming flight in October with Novaspace and ESA STEC. Here on the top right, you see a random positioning machine. So this is something that essentially simulates microgravity. Not really sure we're going to do that because we're running out of time, we're running out of money, and I'm not really sure. I mean, it's changing the vector over time so that the net gravitational force is 0, but you're still being exposed to different vectors and different gravitational paradigms. So I'm a little suspicious of the use of the RPM. But here on the bottom, you can see it's just to show you that on the electromagnetic spectrum at the higher energy level, we're looking at ionizing radiation and then eventually heavy ion radiation to be able to compare a mutational signature to cosmic radiation. And we're using the large diameter centrifuge at ESA STEC on the bottom right-hand corner, and also the 100G centrifuge to look for, again, the impact of different G forces on our samples, but also the mutational signature of the gravitational force. So I think I'm talking quite a lot, 17 minutes. So this is to show you, not only do we have to design our experiment, we have to build our hardware, but then our hardware goes into a space station. For us, it's both the Chinese space station and the International Space Station. We haven't announced this yet, but we do have an ISS mission through Space Application Services, which is a commercial provider. So we're going to actually do a pilot experiment on the ISS before a 10-day mission, before a 31-day mission with China. But in order to send our experiments up into space, we have to build the hardware. And so this is what's available to us. You see payload units here. Here on the furthest left is a payload unit with Space Application Services on the ISS. They literally give you a rack and a cube, but you can imagine with organoids, or any cells for that matter, we have to have a bioreactor that can maintain temperature at 37 degrees, a 5% oxygen or 5% CO2, certain oxygen saturation, imaging, fixation. So not only do we have to design our experiment, we have to actually build the hardware to send our experiment up. So this is what we're given. And then I need to find the money and find the team to design the bioreactor that fits in these little cubes so that my experiment actually works. So here you see a quick pictorial of our partners. Norway's in the center, myself as the principal investigator. We have several European partners, and you can see China. And this is color-coded because Norway is a part of the European Economic Union, but is not a part of the EU. Our partner countries are part of the European Union, and China, of course, is off on its own. And this is just to show you that when we're talking about sending experiments and hardware across borders, there are huge geopolitical lines. There's huge issues around export control. I mean, the paperwork I can tell you on my desk is like, it's not as simple as saying, hey, Norway, here's my experiment. Thank you for designing my hardware. Send it up to space. The hoops, the admin, everything that we have to jump through and get done before we even handle the science is quite extreme. So just to wrap up, I think I'm on time. I wanted to share with you a few lessons learned. Yeah, 10 seconds. A few lessons learned. One, momentum is a habit. And what I mean by that is the process to get your experiment into space from idea to realization is years. It could be a decade. And it's hard. It's lonely. It's long nights, weekends. It's difficult, and it's really, really easy to lose momentum. And what I've learned is momentum is actually a habit. If we keep at it every day, you show up, whether it's 10 minutes, 20 minutes, 5 hours, you show up every day and you keep that going, and that habit keeps the momentum going, which has a huge impact on your project. Collaboration is a skill. I'm a quite friendly person. I like people, I'm interested in people, but to collaborate across borders, across countries, with national, international organizations, people that need signatures, funding agencies across languages and time zones, it's quite a skill. Best intentions do not equal signatures. No matter what people say, it means nothing unless they're willing to sign that piece of paper. You have to be flexible, and at least for tumors in space, a very difficult lesson, geopolitics comes first, administration comes second, and then comes the science. So I'll stop sharing my screen, and that ends my presentation for today. Thank you.
Thank you so much, Tricia. A great overview of the Tumors in Space project. I love to see and hear about the organelles. Takes me back to my master's in tissue engineering a while ago. But it was also great that you managed to share some of these, well, maybe quite extreme challenges you have dealt with concerning funding, for example, and the ones that you mentioned at the end, but also creating and coming up with an extremely relevant experiment while maintaining the feasibility of such an experiment on board a space station. And we've already got a question in the chat box about that, but we'll get to that at the end of the session. Certainly, you've put space genes in a new perspective for me. I'm looking forward to the launch of Tumors in Space. Next, we will be switching to Dr. Francine Garrett-Bakelman. Hi, Francine. Thanks for being with us today. You've worked on the NASA twin study. We could truly say you're an expert in this field. I'm looking forward to hearing your presentation. The floor is yours.
Okay, great. Thanks for the invitation and the kind introduction. And I really enjoyed Dr. LaRosa's talk. So let me start out by saying is that there is absolutely no way I can cover the entire twin study in a 15 to 20 minute talk, okay? So the first thing I'll say is that I'm going to provide an overview. I will highlight a couple of things from the science that was performed. I already put in the chat the website link for the NASA twin studies. So there is a tremendous amount of information there. And some of my slides will include some additional links, which I can also put in the chat to kind of follow up stories that have been published that emanated from this original study, but that have really expanded upon our understanding of the hypotheses that were generated during this time. So I would always like to start by thanking the people that I work with. And this is a busy slide, but I do want to take the time to go through it because without these people, this project would not have been possible. So at the time that this project was being performed, when the science was being performed, I was actually a postdoc at Weill Cornel Medicine. And my PIs, Ari Melnick and Christopher Mason, co-mentored me. Chris Mason is a computational biologist, and he was actually the PI that obtained funding to participate in the NASA twin study. And I was extremely fortunate to be there at the time and for him to approach me to participate with him on this journey together. And then I'd like to thank the Twins Working Group. This was a really very collaborative project. There were 10 different institutions highlighted here that participated in the project. I think ultimately there were 89 authors on the original paper that was published. And And our leaders were really from NASA and other organizations. And I just want to highlight Craig, John, and Graham, who really initiated the process, created the funding opportunity, and really helped us get to the finish line of this twin study, which hopefully is just the beginning of what could be done in space with human biology studies. And of course, to thank the study subjects. So, you know, the twin study was really a concept that emanated from the desire to travel to Mars. And as was mentioned by our host earlier, this is a long-term mission. It's about 3 years. And currently we're only here in an International Space Station and on some of the other space stations that were mentioned by the talk before me. And there's a tremendous amount of engineering and progress that needs to be made for us to make it to Mars and back. But one of the open-ended questions is really what's going to happen to humans? Will humans actually survive this trip to Mars and back? And the twin study was one of earlier attempts to try and understand how longer-term space missions affect human biology. And as many of you probably know, currently, most astronauts only spend between 3 and 6 months on an International Space Station. The missions before the current era were much shorter than that. Most of our trips are, at least now, are only to this area, which is within the Earth's microgravity area. And, you know, very few people have gone to the moon. And so we really don't have a good understanding of the full impact of space travel on human biology. And furthermore, we really have no idea what a 3-year mission would do to a human being, physiology and biology. Sorry, is there a question?
No, sorry, go on. We'll save the questions for the end. Thank you.
Sure. So, you know, the question was, if you take a human and you put them in space for a year, what aspects of biology and physiology change? And the twin study really was a synthesis of a variety of measures. And I do not have time to go over them today, but what I do want to focus on today is more on the genomics aspects of the study that was done. So as most of you probably know, there's the central dogma of gene expression. Gene expression is essential for cells to be functional and to be healthy and to be able to support human life. And you know, I don't want to go through this in extreme detail, but at any level of this process, from genome to physiology, things can go wrong. And the question is, how much errors or abnormalities are associated with space travel that is longer than the standard travel time? Is it reversible? And or are there mitigation approaches that could be applied to help alleviate abnormalities in this system that's really essential for life? So the ISS, as all of you know, is a great place to conduct some of these studies. And as was mentioned before, model systems are very hard to study in space. I think the dogma today is that studying humans is really the optimal way to understand how space travel will affect humans in long-term space travel. But of course, there are many other models, and some are highlighted here, that have been studied in space for many years, and we've learned many lessons from them, but they do have a lot of cons. And the main challenge with the human study is the people and the challenges of conducting it. And a big thing is what do you do about controls for your experiments? So I really admire Dr. Lacroix's approach where, you know, she can really build in some of these issues into her experiments and really conduct well-controlled experiments that you'll be able to learn many new insights from. So the twin study was NASA's first effort in trying to integrate genomics and physiological measures and to study those in the context of a longer term mission, a 1-year mission. And the basic of the study was these are twins, the Kelly twins, and I'm not breaking any HIPAA violations. This is a very, very well publicized study. And they were both astronauts in the past. They both have retired now. But Scott had spent much more time in space, and he was still active at the time that the twin study was conceived. And Mark was retired. And so the idea was that we would compare them over time. Scott would spend a year in space, and we would collect a variety of specimens and physiological markers to do this comparison. And as I mentioned, this was a true collaborative effort. And I'm just highlighting some of the main areas of study that many people participated in throughout the US. And my particular participation focused on the transcriptome or gene expression analysis. However, this was really tied with a number of the other groups, and I'll try and highlight that in the next couple of slides. So in order to collect the specimens for this study, there was a lot of preparatory work, as was described by my colleague earlier. But ultimately, what we wound up doing is we collected blood, stool, and urine samples either on Earth or on the International Space Station. Some were directly processed, some were frozen. If they were on the ISS, they of course had to come back. So they came down in the Soyuz capsule, and then they were flown back to the US. And then some of them were fresh, so they were collected the day that the Soyuz was leaving, and some of them were frozen. And so we had some specimens that were ambient return, so they were never frozen on the ISS itself. All the samples were processed the same way. And I was heavily involved in the process of kind of developing the protocols and on the ISS protocols for sample collections for blood, and then processing the samples that had been stored to do the genomics assays. So our original report was this in 2019. It was a massive effort, as I mentioned, by many people who came together and thought about this all together. But you know, there were interesting findings, but at the end of my talk, I'll kind of highlight some of the shortcomings of the study as well. So overall, we found that there were many changes that were associated with flight. Most of these, however, were within normal limits of variation. And many of these returned to pre-flight states upon return to Earth, either immediately or a couple of months later on. The findings were really hypothesis generating and were estimates of potential risk for long-term missions because we only had an N of 1. But it was really a beginning of the human genomics era of NASA's research and space travel. So it was a really exciting project to be involved with. But I do want to highlight that in 2020, there were almost 30 additional papers published. Some of them used some specimens and data from the original study. Some of them were extensions of the original study. So there's been kind of a continuation of this story over time. And if you're interested, I'm happy to put this link in the chat after my talk. So this is a overview of all the specimens that we had collected. It's a mixture of blood, vasculature, and these are all physiological and molecular collections, pre-flight, in-flight, and post-flight. And like I said, I don't really have time to discuss everything that was found, but I wanted to highlight a couple of examples of findings that we had and some of the hypotheses that we had generated, and hopefully these will be followed up upon. So telomeres, as you know, are very dynamic. They have information in them about biomarkers and determinants of aging in terms of health and disease, in terms of genetic and non-genetic factors such as exposures. And in general, astronauts have been found to have shorter telomeres and lower levels of telomerase activity. Telomerase is the actual protein that regulates the length of the telomeres. And the hypothesis was that with travel to space, there would be accelerated shortening of the telomeres and elevated telomerase activity. And this was led by Dr. Bailey. And to her surprise, the answer was no. Her hypothesis was incorrect. And actually, what we found is that the telomere lengths were prolonged during space time, the flight time, and then they came back to almost pre-flight levels after return to Earth. This is data from our original report. This was a follow-up study that looked at an additional, I think, 59 astronauts that had flown for 6 months, and she saw the exact same pattern that we had seen in the one-year mission, suggesting that this is a true phenomenon, which is really not well understood. Our theories about it is that there might be some stress levels that are causing this to happen, or perhaps some radiation exposures that are inducing this telomere elongation for protection of the genome. But unfortunately, during flight, we weren't able to measure the telomerase activity because the ambient returns took too long for that to occur. And so we were never able to answer the question of whether this elongation that we're seeing was truly because of telomerase activity changes or not. But that's a theory that Dr. Bailey is still pursuing today. But to come back to the question of stress, we and others, so this was from the original twin study, this is from a follow-up study, and there was a significant signature of oxidative stress during space flight. And this was, as I mentioned, shown during the one-year mission and also during these six-month missions, possibly being the culprit for the association with the elongation of telomeres. Now, another interesting finding that we had was that there were quite a few thousand genes that became differentially expressed, and you can look at the middle orange and purple to indicate that during flight when compared to the ground subject. However, interestingly, 93% of these genes returned to pre-flight levels at 6 months after coming back from the ISS. Now, we never measured past 6 months. That was never approved in the original study, but who knows if we had measured things a year after return, whether 99 or 100% of these genes would have returned to normal by that time. We just don't know. But in the signatures that were what we call flight related, there was enrichment for genes involved in the immune system, DNA repair, bone and hypoxia, hypercapnia regulation, which some of these you could maybe expect, but we hadn't had the data before this to really show this genome-wide genomics type of picture that supports those hypotheses. And furthermore, the hypothesis and the data suggesting this inflammatory state induced upon space travel was also orthogonally confirmed in cytokine assays, where we measured their level in terms of inflammatory state. And interestingly here, there were both pre and in-flight associated cytokine elevations in the subject, suggesting that this is perhaps part of the preparatory process that gets enhanced during spaceflight. But there were also definitely return associated cytokine signatures showing a very enhanced inflammatory response upon return to Earth. Some of it lasted past the immediate return period. Some of it was very acute and only lasted a couple of days after return, which was quite interesting. And this has been also confirmed in that additional study that looked at almost 60 additional astronauts. So we feel that our findings from the twin study suggesting that there is an inflammatory state induced during flight is a valid finding. It happens and seems to be in most astronauts that travel more than 3 months. And it's one of the things that could be targeted for mitigation in the future to help prevent any unwanted health consequences. Finally, I just wanted to highlight one more aspect of the study that really focused on physiological adaptation. This looked at protein levels and metabolites over time during the project. And one of the findings was that lactic acid was quite elevated in the flight subject during the flight time. And some of the reasons that this could result from are highlighted here, but the question I wanted to focus on today is on mitochondrial function. So because we had these signatures of inflammatory state from the flight period, the hypothesis was that perhaps this milieu of cytokines and inflammatory state could be affecting mitochondrial function in cells in Scott. And so we obtained plasma from Scott's blood, and we exposed cells in vitro. So it was after specimens were collected and brought back to Earth. And we assessed for mitochondrial function, and I'm not going to have time to go through this assay, but needless to say, if you take plasma from patients that have mitochondrial dysfunction, you can impair mitochondrial function in vitro. And that's exactly what we saw. This was the ground subject, and you can see that all of his specimens, the plasma specimens that were used, didn't induce any mitochondrial dysfunction. But from Scott, the plasma specimens actually induced mitochondrial dysfunction. And this continued post-flight, consistent with the inflammatory state that we detected in the cytokines and the gene expression signatures. This was also associated with detection of cell-free mitochondrial DNA. So not only are the mitochondrial dysfunctional, but they also seem to be getting damaged in some way. So, the conclusions from some of these limited experiments that I showed to you is that there is flight-limited lengthening of telomeres, which is reversible, appears to be reversible. There are space-associated transcriptional reprogramming, which is largely reversible upon return to Earth, and that aberrant mitochondrial function and elevated inflammatory responses are detected during flight. Now, this study is limited. We only had one twin pair. There were no other astronauts that were tested on the flight to this extent in terms of the genomics assays. And there were several environmental factors that were confounders between the twins that we couldn't really control for. For example, the twin on Earth didn't eat, like the food that Scott was eating on the International Space Station. And so the clinical significance of these differences is really unknown that we found between them. They might be applicable as biomarkers to monitor health, Maybe some will be applicable for future interventions to prevent harm. And one of the things is that we did not use advanced research methods that were CLIA certified. So all of these findings will need to be confirmed ultimately in additional studies. Now, the nice thing though, is that towards the end of the study, there were efforts to start to bring molecular biology and sequencing to the International Space Station. And there was actually a nanopore technologies that was tested on parabolic flights that was mentioned before. And then we actually, Chris, my PI at the time, actually helped facilitate getting a nanopore sequencing machine onto the International Space Station. And Kate Rubins, who was one of the astronauts who worked with him, actually sequenced some samples from the ISS and actually got detection of sequences. So it is possible that very soon, some of these studies that we conducted and generated data from molecular biology and some of these follow-up studies here will be able to be conducted directly on the International Space Station. And that's what I'm hoping for, is that, within the next decade, some of these complex issues with generating genomics data and analyzing things more real time will really be transitioned to the ISS and to other space station locations, and we'll be able to learn much more about what impacts human biology in space. So I'll stop here. I'm going to put this link here in the chat for people to explore further. Thank you for your attention, and I'm happy to answer any questions.
Thank you, Francine. A great overview of the NASA Twin study and your research participation in it, in your experiences and sharing some of the results. I also realized that 15 to 20 minutes is too short for this topic. Oh, Siri is talking to me. Sorry about that. As you said, you shared some material in the chat box for our audience if people want to dive in further. I think this was very informative and inspiring for many viewers. Thanks for that. This brings us to the Q&A part of our episode. We have a little bit of time for a few questions. Francine, the European Space Agency is currently looking for new astronauts. I'm one of the applicants myself. You worked with NASA, that's generally more regularly recruiting astronauts. Do you know if NASA is planning to do more of twin studies in the future, or do you think we have still enough data gathered from this one?
There is not enough data. Let's put it that way. Okay, period.
Never enough data.
There's significant interest in having additional studies like this. I think it's very rare to have a pair of twins that you could study in this way. And so, I'm not a NASA representative. I cannot speak on their behalf, but my understanding is that there are efforts to continue to do longer term missions with additional astronauts, volunteers who are willing to stay up there for more than 6 months. And there will be efforts for furthering the genomics assays that, we did to additional people. Everything we saw was within 2 people and from the same genetic background, right? There's no lessons here about variation and what is possible, right? Everything we found may have been a fluke because it's just from one person. I think the follow-up studies are suggesting that some of the key findings were not flukes and that they were real. But many more studies are needed to really prove this more definitively. All
right. Well, since we have limited time and our scope of the scope of the topic is so broad, there can be endless questions about this. So what I'd like to do is ask a final question to you, Dr. LaRose, and maybe Dr. Garrett-Bakelman, if you maybe have something to add. I think many of the participants will wonder or would like to pursue a career like the one you have, and would wonder how you got involved with this field and this work. And so what advice do you have for someone interested in doing research in this field of space health?
So I can share with you, my first degree was physics, and I Googled what to do with a physics degree. And it said, do a master's. And the Canadian Space Agency popped up. And the Canadian Space Agency said, go to the International Space University. And that's what I did. And that was many years ago now. So I think ISU for me, the International Space University, was a real cornerstone in my career. Thank you.
Great to hear. Francine?
Yeah, and I would add, as somebody who's an outsider, is that, you know, if you're really interested in this, opportunities to work with people who are engaged in this type of work do exist. Sometimes they're hard to identify. However, the space agencies, I'm sure the Europeans, I know NASA has this too, they have training programs for postdocs and graduate students. And I know that sometimes there are opportunities for young faculty interested in the field, but don't have like the exact background training for it, to kind of take opportunities like that and explore them very seriously, if that's something that you want to pursue. Great. It's not something I did. I think I just got really lucky. I was at the right place at the right time. And it was like a dream come true for me, because I had considered going into space medicine as my medical training, and I decided not to. So this was like a second chance for me, and it was just awesome. And I'm just so happy that I had that co-PI mentor who called me up one day and say, hey, do you want to work with me on this project? And I was like, do you even have to ask? It was like, great. It was awesome.
Wonderful. Wonderful. Thanks for sharing that. We are at the top of the hour, which means that we have to conclude this first episode of the second season of the Health in Space series. I'd like to give many thanks to our speakers, Dr. Tricia L. LaRose and Dr. Francine Garrett-Bakelman, for being with us today. If you would like to know more about tumors in space or the NASA Twin study, feel free to reach out to them. For now, the presentations you gave us were fantastic. You gave us a broad understanding of your expertise in the space life sciences and all the challenges that comes with them. Thanks for taking the time. And to our audience, if you liked this episode, check out the SGAC YouTube channel for more previous episodes, other materials, and feel free to share them on social media. This was the start of a new season, but we are evaluating new topics for new episodes. We have a whole list of fascinating topics that come to mind when thinking about health in space. But if you have a topic that you're passionate about and want to learn more about, find us on LinkedIn, Twitter, or email and let us know. We'd like to create this series with you together, our audience. That was it for today. Thank you again, Dr. Tricia LaRose, Dr. Francine Garrett-Bakelman. Thanks for everyone that joined us from around the globe, and have a great day ahead. See you next time.